So in this video, we're going to talk about viruses and prions. So we're going to start by talking about general characteristics of viruses. And the first, and one that we've talked about since the beginning of class, is that viruses are not considered to be living. And that's because they don't fill the characteristics that we define as life. They are obligate intracellular parasites, which means that in order for viruses to replicate, they have to be able to infect a host cell, such as a bacteria, a protozoan, fungi, algae, plants, or animal cells, and they turn those host cells into virus-producing factories. And so they can't replicate on their own. So that's why we call them an obligate intracellular parasites. In terms of viruses, remember that viruses are ultramicroscopic, meaning that you can't see them with a typical compound light microscope. If you remember for E. coli are about 1 micrometer roughly in size, 1 to 2 micrometers. And in terms of nanometers, that would be 1,000 nanometers. And so notice that if we look at the size for viruses, they range from as small as 20 nanometers to 450 nanometers. And so they are much, much smaller than bacteria typically. They are, again, not cellular in nature. Their structure is compact and economical. They don't carry a lot of their own enzymes. Some viruses may have a few enzymes, some might have none at all, and that's because, again, they rely on the host cell in order to replicate and turn the host cell into a virus-producing factory. When we talk about the structure of a virus, viruses typically are made of a protein shell or a capsid surrounding a nucleic acid core. And we're going to talk about that nucleic acid, but the nucleic acid in viruses varies quite a bit. Typically, they're either DNA or RNA, but not both. That's a general rule. There is an exception to that rule, but as a whole, either the virus contains DNA or RNA, but not both. In terms of their structure of nucleic acids, they could be double-stranded, single-stranded, DNA, RNA, circular, linear. It ranges quite a bit for viruses. We're going to talk about that on the surface of viruses, they have molecules that basically give high specificity for attachment to host cells. And we're going to talk about what makes a virus able to infect some cells and not others. And again, they have to basically take control of the host machinery and basically turn themselves into a virus-producing factory. And again, that's because they lack most metabolic processes or enzymes, and they lack machinery to synthesize their own proteins. So if we look at the size of viruses, so here's an approximation. So let's say we're looking at a human red blood cell. Here we're looking at E. coli. And so notice that E. coli are about, again, 1 micrometer in going across. And again, that is equal to 1,000 nanometers. If we look at viruses relative to both the red blood cell and the E. coli, what you'll notice is that the viruses range in size, yet they're much, much smaller. And again, that's because we know this because we have to have an electron microscope in order to visualize them. So if we look, so here's the Ebola virus, which causes hemorrhagic fever, which we'll talk about in a little bit. It's about 970 nanometers. If we look at a rhinovirus, rhino like the nose, rhinoviruses are about 30 nanometers. So notice that they're very, very small, yet they cause the common cold. Ebola is much larger and it causes hemorrhagic fever, which patients typically die from. And so what it's important to notice is that it's not the size of the virus that matters. Virus particles are infectious and cause various diseases independent of their size. So there's no correlation whatsoever between the size of a virus and how damaging it is. When we talk about a virus, we often will talk about a virion. And a virion is a fully formed virus that's able to establish an infection, meaning that the virus is assembled and it's ready to infect a host cell. So, which of the following is true regarding viruses? Red, virions contain both DNA and RNA. Yellow, they multiply by binary fission. Green, they're sensitive to antibiotics. Blue, they are not composed of cells. So take a minute, think about your answer, pause the video, and when you're ready, go ahead and turn it back on. So if you said blue, you are correct. So viruses, remember, are obligate intracellular parasites. So they are not composed of cells. Red is not true. Virions contain both DNA and RNA because remember we said that for viruses, it should be that they contain DNA or RNA, not both. Yellow says they multiply by a binary fission. Remember that this is the way that bacteria reproduce. And so this is not true. Viruses have to replicate within a host cell. It's not green. They're sensitive to antibiotics because remember we talked about that for antibiotics. Antibiotics are specific to bacteria and they have no effect on viruses. And so you can't take an antibiotic for the common cold. So the way that viruses are classified is based on a couple different things. The first is the type of nucleic acid. Do they use single-stranded RNA? Do they use double-stranded DNA? What type of nucleic acid do they have? They're also classified by their morphology, which remember means their shape. Are they helical? Are they polyhedral, which we'll talk about in a minute? And lastly, they're also classified by their replication strategy, meaning that the way that they replicate within the host cell. So we're going to start by looking at viral structure. So if we look at a virion, or the virus particle, we can break down the structure by two parts. One would be the covering, or the outer part of the virus, and all viruses have a capsid. And we're going to see in a minute that this is this protein that's found on the outside of viruses. Some viruses, so some, have what's called an envelope, which is a membrane structure, but this is not found in all viruses. Some have the envelope, others that lack the envelope, we're going to see, are called naked viruses. And so this is only found in some. Inside the capsid, we're going to have our central core. And the central core has the genetic information. So that's the nucleic acids. Again, it can be DNA, it can be RNA, it can be single-stranded, double-stranded, etc. But they have some sort of nucleic acid. And some viruses have these matrix proteins. And we're going to talk about some of the enzymes that some viruses do have. But again, this is not true for all viruses. So if we look at the structure of the viruses, we have a naked nucleocapsid. And what that means is that it lacks an envelope. So it has its protein capsid, it has its nucleic acids, and that's pretty much the structure. Other viruses are what we call enveloped viruses. So they still have the nucleic acid, they have their capsid, but external to that, they have a structure called an envelope. And this envelope is a phospholipid membrane. And this phospholipid membrane is derived from the host cell membrane. As the virus leaves the cell. And so we're going to talk in a little bit about the function of that envelope. But what I want to mention now is that the presence of that envelope is going to have an effect on whether the virus is more resistant to antimicrobials or less. And so remember when we talked about antimicrobial chemicals, that when we talked about the least resistant, meaning the most sensitive to chemicals, would be the enveloped viruses. So these are going to be the most sensitive to antimicrobial chemicals. And that's because when we look at these viruses, because of this phospholipid membrane, a lot of antimicrobial chemicals are lipid soluble, meaning that they have the ability to dissolve that membrane. And when they dissolve the envelope, the virus particle no longer functions. And that's because you're going to see that in the envelope there are these spikes which are necessary for the virus to attach to a host cell and to get in and replicate within the cell. And so these enveloped viruses are much more sensitive to the antimicrobial chemicals. So some examples of enveloped viruses, this would be like influenza virus, which you can guess causes the flu, varicella virus, which causes chickenpox, etc. And for naked viruses, examples of naked viruses which have only a protein coat, and some examples of this would be like rhinovirus, which again causes the common cold. We have papillomavirus, which causes genital warts, for example. And so again, these are more resistant to antimicrobial chemicals. And so we're going to look at some more detail about these two types of virus particles. So nucleocapsid is found in all viruses, and it has two main parts. It has a nucleic acid core, and it has this protein capsid. And so we're going to talk a little bit more about each one. So first we'll talk about the nucleic acid. And again, the viral genome is either DNA or RNA, but never both, again, as a general rule. The genome can be single-stranded, double-stranded, it can be linear or circular. And so the viral genome really varies a lot. In some cases, the viral genome may also be segmented, meaning that the individual virus particles might not contain the entire genome. It might be that the organism has to be infected with multiple parts in order for the viral genome to be complete and in order for the virus to be able to replicate inside the host cell. And so the genome of viruses really is a very diverse group. The nucleic acid carries the genes necessary to invade the host cell, so it allows them to get into the host cell. And it also has information that allows the virus to redirect the cell's activity to make new viruses, basically meaning that it allows the virus to be able to hijack the host cell machinery. The number of genes varies for each type of virus. They can have few to hundreds of genes depending on the type of virus. So if we look at the capsid is found in all viruses. And again, it's this protein coat that encloses and protects their nucleic acids. Each capsid is constructed from identical subunits called capsomeres. And these capsomeres are made of protein, and they are these repeating subunits. And so, if we look at these capsids, remember that some viruses are naked, meaning that they just have a capsid on the outside. Other viruses, remember, have an envelope, which is a structure that is external to the capsid. And so we're going to talk about what does an envelope do for a virus. So, for enveloped viruses, these are only found in some. Again, otherwise they would be called naked. And typically, the types of viruses that have an envelope are going to be mainly animal viruses. And that's because this envelope surrounds the capsid, and the way that the envelope forms is that after the virus infects the host cell and turns that host cell into a virus-producing factory, when the virus leaves, it sometimes does something called budding, meaning that it actually buds off from the host cell. And when it does that, it takes some of the host cell membrane with it. And so when it takes that membrane then is derived from the host cell itself. And so one of the purposes or one of the functions of this envelope is to hide from the host cell, the host's immune system. And that's because many of the antigens on the outside of this virus particle are derived from the host cell itself. And so the immune system recognizes it as self and doesn't attack it. And so the function of the virus, the envelope, is to hide from the host immune system. The envelope has these exposed carbohydrate protein complexes on the outside, and these are what we call spikes. And you're going to see in a minute that the spikes are essential for attachment of the virus to the host cell. They are what give the specificity for a virus to be able to infect a particular cell type. When we look at enveloped viruses, they can be pleomorphic. And remember, pleomorphic means many shapes. And that's because this membrane or envelope on the outside is a very fluid structure. And so these envelope viruses have many shapes. So for the function of the capsid and the envelope, both are used to protect nucleic acids when the virion is outside the host. Basically, again, think of it almost analogous to an endospore, right? That the outside structures are there to protect the genetic information. Additionally, the outside components of the virus helps the virion to bind to a cell surface, and this allows it to assist in the penetration of the viral DNA or RNA into a suitable host cell. And so again, it's those components on the outside of the cell that give a virus specificity to its host. Now, remember that I said that viruses have few to no enzymes for their own metabolism. For example, they lack enzymes for protein synthesis and ATP generation. In order for viruses to multiply, they must be able to overtake the metabolic machinery of the host cell. However, some viruses do contain a few enzymes, and some of these are examples. And so the first is going to be the polymerase. Remember when we talked about DNA replication, polymerases are used to synthesize new DNA or RNA. So if they're going to make DNA, it's called DNA polymerase. If they're going to make RNA, it's RNA polymerase. There are also enzymes called replicases, and these are used to copy RNA, meaning that in this case we go RNA and replicate it to RNA. And remember that this is different because for polymerases, when they make RNA, typically they're going to go from DNA to RNA in what we call transcription. Now, some viruses, like HIV for example, have an enzyme called reverse transcriptase, and these are used for synthesis of DNA from RNA. And so again, it's going in the opposite or the reverse direction of transcription. And so we're going RNA going back to DNA. And so notice that in this case, reverse transcriptase allows viruses to go RNA to DNA. And this is used for viruses that have their genetic code as RNA. So used for viruses that have RNA as their genetic information. And again, an example of this would be HIV. And so some of the drugs that are used to target HIV are used to inhibit reverse transcriptase. Because if we block reverse transcription, then the virus can't take their RNA, their genetic code, synthesize back to DNA to turn the host cell into a virus-producing factory. The last enzyme is going to be what's called lysozyme. And this is an enzyme that's used to degrade cell walls. And you're going to see in a little bit that when we talk about the immune system, actually your own body also produces lysozyme, tears, for example, mucus, and that lysozyme is used to basically degrade cell walls. And if you remember back to which types of pathogens would have a cell wall, remember that in that case, it could inhibit bacteria. And so having lysozyme in your body is part of your non-specific immune response. In viruses, some viruses have lysozyme to degrade cell walls, and this is found in bacteriophage, which remember are viruses that infect bacteria. Sorry, let me make that G look a little better. And so in virus particles that are going to infect bacteria, they have to have the lysozyme to break down or degrade the cell wall so that they can inject their DNA into the host bacteria. So if we look at the morphology of viruses, the capsid shape results from different capsimer arrangements. And so there are four main types of morphology. The first is going to be what we call a helical virus. So here's an example of a helical virus. And this is going to be a hollow cylinder with nucleic acids inside. And so an example of this is Ebola. And you probably remember hearing about Ebola in the recent outbreak. And remember that Ebola is a virus that causes hemorrhagic fever. If you've ever seen the movie Outbreak, that movie is in reference to Ebola causing disease. And in hemorrhagic fever, the organs begin to kind of turn to mush and patients start to bleed out of their orifices. So they might start bleeding blood out their ears, their nose, etc. Another example of a helical virus would be like rabies, for example. If we talk about a virus being a polyhedral, that means that that virus is many-sided, right? Remember that poly means many. And in this case, they're usually 20-sided. Sided, which we call an icosahedron. An example of a icosahedron would be like an adenovirus example. And an adenovirus causes respiratory infections. We also can have complex virus morphology. And in this case, an example of this would be like bacteriophage. And a bacteriophage is going to have a structure where it has the capsid that is an icosahedron, and this tail sheath is actually helical. And so notice that in this case, this virus has both types of morphology. And so that we call a bacteriophage. And that is going to be a complex virus. And lastly, we have what's called an enveloped viruses. And in an enveloped virus, these are roughly spherical, but are polymorphic, meaning that they can have many shapes. And an example of this would be like influenza virus. And when we look at the capsid inside of an enveloped virus, the capsid inside can either be helical or polyhedral. So, question for you. Capsids are composed of protein subunits known as, is it red, capsimeres, yellow, spikes, green, envelopes, or blue, tail fibers? So pause, and when you're ready, go ahead and push play again, and you can get the answer. So if you said red, capsimeres, you are correct. The subunits that are repeating in a capsid are referred to as capsimeres. Some viruses do have envelopes, and those envelopes, though, are not made of protein subunits. They're made of lipids instead. On envelope viruses, you can find spikes. Spikes can be made of proteins, but that's not a part of the capsid. That's actually part of the envelope. And you're going to see later that the tail fibers are what are found in viruses like bacteriophage. So now we're going to look at how an animal virus multiplies. So there are 6 general phases in an animal virus multiplication cycle. And the first stage is going to be adsorption or attachment. And this is where the virus binds to specific molecules on the host cell. And we're going to talk in a minute about how, depending on the cell surface markers on the host cell, that will dictate which viruses can infect which cell types. And that's something called host range. And what you guys are going to see is that some viruses have a very wide host range, meaning that they may be able to infect multiple cell types or in multiple organisms. Others have a narrow host range, meaning that they might only be able to affect one cell within one particular species. And so we'll look at that in a minute. The second phase is known as penetration, and this is where the virus penetrates the cell, meaning that it gets taken in to the host cell and the genome gets in. Stage 3 is looking at uncoating. And in uncoating, the viral nucleic acid is released from the capsid, meaning that in this stage, this is where the protein capsid gets broken down and the nucleic acid is released into the cell to begin to make new virus particles. And so you can think of uncoating kind of as like taking off its coat before it comes inside. So it has to remove that capsid to expose their nucleic acids. The fourth stage is going to be synthesis, and it's just like it sounds. The viral components are produced, meaning the cell is going to replicate its nucleic acids, it's going to make its capsimeres, it's going to make all the components that it needs to form new virions. Step 5 is going to be the assembly, and this is where the new virus particles are constructed. And so at this point, now the cell is going to put together the virus particles to form the functional virions. And then the last stage is going to be release. And this is where the assembled viruses are released from the host cell. And we're going to talk about that there are two main ways that animal viruses can be released. One is referred to as budding or exocytosis, meaning that in this case, the virus particle leaves by just taking part of the host cell membrane with it. And when this happens, it doesn't necessarily kill the animal cell right away. The second option for release is what's called cell lysis, meaning that in this case, it causes the host cell to rupture or break open, and the virus particles are released and ready to infect another cell. And so we're going to look into more detail for each of these. And so when we're looking at the multiplication of an animal virus, you can see these steps now in pictures. So again, first we have adsorption. The virus particle is going to attach to receptors on a particular host cell. Then we have penetration. Now the virus is penetrating, it's getting taken into the cell. In this case, it's using what's called receptor-mediated endocytosis. And so now the virus is getting taken into the cell. Then we have uncoating, and in this case, the envelope is removed as well as the protein capsid. Now the nucleic acids are exposed, and now the virus can take its genome and direct the host cell to making it a virus-producing factory. And so now we have synthesis, and this is basically where the cell is going to synthesize the new components. It's going to make the new RNA, it's going to make the new capsimeres, it's going to make the new spikes, and after it makes all the parts, then it has to put them all together. And putting it all together is in the assembly stage. So now it's going to start to assemble the productive virions. And then lastly, we have release. And in this particular case, we're looking at an enveloped virus. And so the way that it's removed from the cell is through simply budding. It's going to take part of the host cell membrane with it. And now we have our functional virion that's ready to go and infect an adjacent cell. So when looking at absorption, in order for a virus particle to be absorbed or to be attached to the host cell, the virus has to randomly collide with a susceptible host cell. And so remember that molecules have molecular motion, and so if a random virus happens to collide with a host cell that has those receptors, that will allow the virus to be absorbed or taken in to the host cell. And so these receptors are found on the host cell membrane. And this is determined by the availability of appropriate attachment sites on the surface of the host cell. if there is no appropriate receptor or if there's a mutation in that receptor, then the virus can no longer be attached to the host cell and is no longer going to infect that host cell. And so if you remember, we talked about for HIV that some people are naturally resistant to HIV. And the reason for that is that on those people who are resistant, they have a mutation in their receptor on their T cells. T cells are the type of cells that HIV infects. And patients who are immune or resistant to HIV, their mutation affects the receptor so that HIV can no longer bind to the T cells and no longer infect those T cells, or not no longer, but just can't infect those T cells. And so as a result, if HIV cannot infect those cells, then the patient can't be infected with HIV because HIV needs the T cells in order to replicate. And so that's why some people are naturally resistant to HIV. Now, some viruses might be able to infect multiple cell types or multiple species, but most viruses can only infect one host species. And so the spectrum of cells that a virus can infect is what we call its host range. Some viruses have a narrow host range, meaning that they might be able to infect only one species, and even within that one species, maybe only one cell type. An example of this would be hepatitis B. Hepatitis B only infects human liver cells, and so this we would call narrow host range. Additionally, poliovirus has a little bit more broad of a host range, and now it's not only infecting human cells, but primates. So humans are a type of primates. Monkeys, for example, would be considered a primate. And in this case, poliovirus can infect primate intestinal and nerve cells. So now we're getting a little more broad. Rabies, on the other hand, infects various cells of many mammals. So typically when you think of rabies, you think of dogs, bats, etc. And this virus not only infects just one species, it affects multiple species and multiple cell types. And so this we would say has a broad host range. So when we look at absorption always occurs via receptors, meaning that the host cell has to have these receptors available that the virus can bind, which will allow the virus to be taken into the cell. Now, the way that the virus is absorbed is that it's a little bit different if we're talking about an enveloped virus or a non-enveloped or naked virus. In both cases, they have receptors, and these receptors are usually proteins or glycoproteins, which remember glyco means sugar, proteins means proteins. So they are these receptors that are a mixture of protein and sugar. And in enveloped viruses, the way that it is absorbed is that the virus uses spikes located on the envelope to bind to receptors that are found on the host cell. And so here are the host cell receptors. The virus has its envelope spikes, and in order for the virus to penetrate and to be able to get into and attach to the host cell, these spikes have to bind to the appropriate receptor. On the other side of this are naked viruses, otherwise known as our non-enveloped viruses. The attachment sites are small fibers on the corners of the icosahedron. But again, notice that whether we're talking about a naked virus or an enveloped virus, both of them have these attachment fibers, whether it's an envelope spike or just these small fibers. Both of those need to attach to receptors, and when they attach to the receptors, that allows them to be attached and to be adsorbed. An example of an enveloped virus, this would be like influenza. And again, in terms of a naked virus, this would be like a rhinovirus. So, once the virus attaches, then entry needs to occur. And that means that the virus particle needs to be able to get in to turn the host cell into a virus-producing factory. And there are two main ways that this can happen. One is through something called endocytosis, and specifically it uses receptor-mediated endocytosis. And what happens in endocytosis is that molecules on the virus attach to specific receptors on the host cell membrane. And so what happens is, in this case, once this binds, that's going to trigger the cell to undergo engulfment, meaning that the host cell is going to kind of pinch in and it's going to bring in the virus in a vesicle. And once that happens, then the virus has to go through the third stage, which is the uncoating. And in uncoating, the envelope has to break down, as well as the protein capsid, to release the DNA to be able to make new virus particles. And again, think of uncoating like taking off a coat. It's taking off the external structures to release the nucleic acids which are inside. On the other side of this is going to be what we call fusion. And in fusion is going to be where the host cell has receptors and it binds to molecules on the virus itself. And in this case, the receptors and the spike complexes will fuse, and in this case, instead of the entire envelope coming inside the host cell, instead it's going to fuse, and what happens is that the capsid is going to be released into the cell. So notice that in fusion, the host cell envelope is not actually incorporated inside the cell. It actually will fuse with receptors on the membrane, and the nucleocapsid will enter the cell. And what happens is now they can have uncoating, and it's going to take off its protein coat, release the capsid, and now we're going to have the free nucleic acids. And so one of the things that's interesting to note is that when cells do fusion, what ends up happening is that when it does fusion, it leaves proteins on the surface of the cell. And this signals to the immune system that something is wrong. An example of this would be for the herpes virus. The herpes virus, the body starts to recognize that the cell is virally infected, and the immune system starts to destroy the cells, and that's when you get cold sores. And so, for envelope viruses, they can do either endocytosis or in some cases fusion. And again, the difference being that in endocytosis, the envelope gets taken in, whereas in fusion, it does not. So we're going to skip talking about the synthesis step and assembly, and that's because depending on the type of virus, there are many, many different ways that synthesis can occur. And due to the sake of time, we're not going to have time to talk about all of those mechanisms. But remember that viral genome can either be RNA, it can be DNA, it could be single-stranded, double-stranded, circular, linear, segmented. There's lots and lots of different ways that nucleic acids can be found in viruses, and this affects the way that synthesis occurs. And so it would take too much time to talk about all of the different ways that synthesis occurs. So we're just going to not, due to time, we won't be able to talk about that. But we will talk about the last stage, which is going to be the release of the virus. And so what happens is that those assembled virus particles need to leave the host cell in order to infect adjacent cells. And so there are two ways through which those assembled viruses can leave. The first is going to be budding, which is exocytosis. And in budding, the nucleocapsid binds to the membrane and it pinches off and it sheds the virus gradually. And in this case, when we're talking about budding, the cell is not immediately destroyed. And so this allows the virus to use that cell over and over to continually make new virus particles. That's in contrast with lysis. Lysis is more common with non-enveloped and complex viruses. And in lysis, they don't just pinch off from the host cell. In this case, the cell actually ruptures or lyses, and this then releases those virus particles, and then those virus particles can go on to infect adjacent cells. Now, notice that for an enveloped virus, envelope viruses would use budding. And that's because, remember that the envelope is derived from the host cell membrane. And so how does it obtain that host cell membrane? Again, it buds off and it takes the host cell membrane with it. So budding would be used for enveloped viruses. The number of virus particles released is very variable depending on the virus. For example, if we're talking about pox virus, about 3,000 to 4,000 virions are released by pox virus. Whereas if you're talking about poliovirus, greater than 100,000 virus particles are released through the poliovirus. And so that can change depending on the type of virus. So this is looking at budding. And so again, notice that here we have synthesis occurred, we have nucleic acids, we have the capsids, we're going to have assembly. So now the capsid and the nucleic acids get put together. And then the end of this diagram is looking at the budding. And in the budding, again, what's going to happen is this is used for enveloped viruses. And so one of the things that happens during assembly is that in addition to making the capsid and the nucleic acid, the virus is going to direct the cell to put the viral spikes on the host cell membrane. And so notice that now you see these viral spikes, and what's going to happen is this nucleocapsid is going to undergo budding. It's going to take some of the host cell membrane with it, with the spikes, and now we have these free virions which are ready to go infect adjacent cells. And so again, in this particular way, this is not going to necessarily kill the host cell. It's just going to keep using the cell over and over again as a virus-producing factory. And when this virus leaves, it's going to take some of the host cell membrane with it. And remember that the purpose of that is to help that particular virus to hide from the immune system. So next we're going to look at how virals are identified, diagnosed, and treated. So in terms of growing animal viruses, remember that for viruses, they're obligate intracellular parasites, meaning that they can't replicate outside of a host cell. So, in order to grow or replicate animal viruses, there are one of three mechanisms that are usually used. The first is to inject the virus into living animals, and the living animals then are used to replicate the virus. The second mechanism is to use embryonated eggs, and so the virus is injected into those eggs, and the viral growth is actually signaled by changes or death in the embryo, and that's because the embryo was turned into a virus-producing factory. Lastly, we have what's called cell culture, meaning to grow certain cell types in a dish. And what happens during this is that these tissues, these specific cell types, are treated with enzymes that allow them to separate cells, meaning that we get isolated individual cells. Then viruses are put on the cells, and we can look for virally infected cells by looking for the cell's deterioration, what's known as the cytopathic effect, or CPE. And so we're going to look at what are some of these CPEs, or the cytopathic effects, that allows us to identify if a cell is virally infected. And during this type of mechanism, we use what are called continuous cell lines, meaning that these cells that are used to be infected are able to reproduce over and over again. And so these cells will allow a researcher to make a large amount of virus. So, what are these cytopathic effects? Well, again, these are virus-induced damage to cells, and this happens in several ways. One is that you might be able to detect a change in size or shape. So notice that if you look at these fibroblasts here, fibroblasts are these dermal type cells. They're kind of long and spindly. If they're infected with a particular virus, instead of having this shape, now they're more circular in shape. And so looking at the overall shape or morphology of the cell usually can indicate whether a cell is virally infected. In addition, inside the cell, we might find cytoplasmic inclusion bodies, or these little vesicles inside the cell. You might see nuclear inclusion bodies, so again, these vesicles inside the nucleus. Cells might fuse to form multinucleated cells. So instead of seeing typically one nucleus within one cell, in some cases the cells actually fuse, and if you look at one particular cell, it might have 2, 3, 4 nuclei, etc. Another effect to host cells can be detected as cell lysis, meaning that the host cells start to rupture. We can have alteration of DNA, so the DNA might be altered based on the infection. And lastly, this can be detected because viruses can transform cells into cancer cells. And so we're going to talk in a little bit about oncoviruses, or viruses that make cells more susceptible to cancer. In order to identify particular viruses, we use several methods to identify the virus. One is to look at simply cytopathic effects. So to look at, are there changes to the cell, again, through the mechanisms we looked at before, either size, shape, looking for certain proteins, etc. So looking at these cytopathic effects. The next way that viruses are identified is through serological tests. And this is the most common way to identify viruses. And so in serological tests, there are several ways through which this can happen. One is what we call a Western blot. And in a Western blot, we can actually detect it by using antibodies. Antibodies are proteins that recognize specific molecules, and so we can see um, if that particular patient, for example, produces antibodies. If they produce antibodies against a particular type of virus, that typically would indicate an infection. Or we could look at proteins to see if a particular virus is found. And so serological tests are the most common way that viruses are identified. The next one is going to be the nucleic acids. So looking for the presence of specific sequences that are typically found in viruses. One of these ways is something called RFLP, or restriction fragment length polymorphisms. So looking for these signals or these sequences that have these slight changes that vary between people or between viruses. We can also use something called PCR. PCR stands for polymerase chain reaction, and you can think of PCR as a DNA Xerox machine. And so the way that this works is we can amplify and detect viral DNAs using PCR. So looking for particular viral DNA sequences. So in terms of treatments can be broken down into several modes of action. The first could be that some drugs actually prohibit or block entry into the cells. So if we talk about entry inhibitors, they inhibit either attachment, they inhibit fusion or uncoating. Basically, again, they prevent the virus from getting into the cell. An example of this would be what's called Tamiflu. Tamiflu is used to stop the action of influenza neuraminidase, which is required for the entry of virus into the cells. And so if you've ever had the flu and you've gone to the doctor, they can give you Tamiflu if you're in the beginning of the infection. And notice that the reason you can't give it too late is because if you wait, then the virus has already entered the cells, and so blocking entry is not going to be effective. However, if Tamiflu is taken early enough during the flu, it can prevent additional cells from being infected by the virus. And so that's the way these entry inhibitors work. We also have nucleic acid synthesis inhibitors, so basically inhibiting the production of nucleic acids. These might include inhibiting viral polymerases, inhibiting reverse transcriptase, again for like HIV, what's called integrase, basically blocking nucleic acid synthesis. Some examples of this, acyclovir. Acyclovir is used for a herpes outbreak, and this works by terminating DNA replication in herpes viruses. AZT is a type of drug used for HIV, and it is used to inhibit reverse transcriptase. And so by inhibiting reverse transcriptase, the virus no longer can go from RNA back to DNA and can't continue to replicate and produce new virus particles. Some antiviral drugs inhibit assembly and release. They might inhibit protease, and protease is an enzyme required to clip or cut viral proteins into functional pieces. And so without that ability, those viruses can't be assembled and released. An example of that is saquinavir or fortovase. And the way that this drug works is that it interferes with protease and virus particles, and these are defective and unable to infect adjacent cells. Tamiflu, in addition to blocking entry, is also used to inhibit influenza virus assembly and budding. And so these are the main ways through which antiviral drugs typically work. So interferons can be used as a treatment, and this is used to prevent the spread of viruses to new cells. And so we can use something called alpha interferons, and alpha interferons are used for viral hepatitis. And that's because interferons are produced naturally by virally infected cells. And when these are produced by virally infected cells, the purpose of your immune system's function or your cells' function for producing interferons is basically to signal cells around it to prevent the virus from being able to replicate within adjacent cells. And so by treating patients with interferons, what that basically does is that it magnifies an immune response. And so that helps the immune system to fight off those virally infected cells. And so this is used to inhibit viral replication and cancer cells also. And again, it's used to basically magnify an immune response, to get the immune system to affect those virally infected cells. And so imiquimod is a drug that's used to promote interferon production. And so again, that's just used to amplify an immune response. So in order for a virus to multiply, several things have to happen. First, it needs to invade the host cell. Again, we talked about attachment, and so in order for the virus to replicate itself, it has to be able to get in. So that's the first thing. The second thing that has to happen is that the virus needs to take over the host's metabolic machinery, basically to hijack the host cell to turn it into a virus-producing factory. And so we're going to talk about three main types of viral infections. And the first is the one that you're probably most common with, and that's what's called an acute infection. And in an acute infection, it's characterized by rapid onset of the disease, meaning the disease comes on quickly, a relatively brief period of symptoms, so typically, again, days, we're not talking years in this case, and resolution of the infection within days. It's usually accompanied by early production of infectious virions, and then it's eliminated by the host immune system. And so notice that the virions get produced and then released from the cell or broken down over time. And so this is an acute infection. It comes on quickly, but it also resolves. That is in contrast to what are called latent viruses. And in a latent virus, what happens in a latent viral infection is that in this case, the virus remains in the cells, but they are asymptomatic. So in this case, they're just kind of hiding out and they're not typically released. However, due to particular stimuli, they may change, and those stimuli might cause then the infectious particles to be produced, and it may reactivate the virus. And so some examples of this, cold sores, which are caused by the herpes virus. If you know anybody who has cold sores, the herpes virus can remain in the host cell throughout the life of the individual. And if the immune system is immunocompromised, for example, like in AIDS, the herpes virus might be reactivated and the infection could potentially be fatal. Herpes simplex virus can hide in the host's what are called trigeminal nerves, so these particular nerve cells, and they might not cause any damage to those cells most of the time. But let's say that the patient then has a sunburn or a fever. That stimulus could reactivate the virus to begin to produce the virions. Some people never show symptoms, meaning they don't even know that they carry human herpes virus. Only about 10 to 15% of people carrying the virus actually exhibit the disease, meaning that they have symptoms. Another example, in addition to cold sores, is going to be shingles. Shingles is caused by varicella virus, and this virus is known to cause chickenpox. And so what happens is that when a patient is infected by this virus and they get an outbreak of chickenpox, the virus gains access to the skin via the blood. And from the blood, some viruses may gain access to nerves and become latent. And so later changes in the immune system, or the T cells, can reactivate these latent virus particles, causing shingles. And so in this case, the virus is kind of hanging out once you've had chickenpox. And if your immune system is compromised, for example, in elderly patients, that can cause the virus to activate and start producing virus particles. And so what ends up happening is that patients get this very, very painful rash called shingles after a chickenpox infection many, many years later, because as their immune system starts to break down, the virus can reactivate, and these are found in the nerves, which is what causes shingles to be a very painful rash. Shingles happens in about 10 to 20% of people who have had chickenpox. For many of you who were vaccinated against varicella, will probably never have shingles, and that's because you don't have the virus latent hanging out in the nerve cells. There are vaccines available for shingles, so to try and prevent shingles from happening later in life. But again, this is an example of a latent infection. A persistent viral infection occurs gradually over a long period. It's generally fatal. And what happens is, in a persistent infection, the difference being that virus particles are made continuously over a long period of time. Notice that if you look, you can see the virus or the virion number increasing slowly, and that's in comparison to the latent infection. In a latent infection, no virus particles, again, they're like in hibernation, so no virions are produced, but in response to some stimuli, it might reactivate that infection so that the body produces, or the cells produce those virus particles. Whereas persistent, we just make them slowly over time. And so an example would be the measles virus. The measles virus can remain hidden in the brain cells for many years, and it slowly increases the number of virions, and it can cause something called encephalitis, or swelling of the brain, and sometimes this can be fatal. Human papillomavirus. Human papillomavirus is an example of a oncovirus, which we'll talk about in a minute. You probably know human papillomavirus as HPV, and that's the cause of genital warts. Now, what you might not think about is that this particular virus causes warts, but also leads to an increased risk of cervical cancer. And so that would be a persistent viral infection. It's producing low amounts of virions for a long period of time until eventually it might kill the patient. So in some cases, some animal viruses enter host cells and permanently alter its genetic material, resulting in cancer. And this leads to a transformation of the cell. These transformed cells have an increased rate of growth, alteration in number of chromosomes, so either the structure or the number, and the capacity to divide for infinite periods of time, which might result in tumors. And so when we look at these virally induced cells, they might have an abnormal number of chromosomes. Instead of having the normal 46 chromosomes, maybe they have 58. Maybe the sequences have changed. But basically, it's altered those cells in such a way that they might grow out of control. Mammalian viruses capable of initiating tumors, again, are what we call oncoviruses. Again, an example, human papillomavirus or HPV has been shown to have an increased risk for cervical cancer. Epstein-Barr virus can cause Burkitt's lymphoma. Hepatitis virus can lead to liver cancer. And so, in this way, these viruses actually can initiate or can help cause tumors, which is why we call them oncoviruses. Think of like a doctor for cancer is called an oncologist, right? So onco refers to cancer. So oncoviruses are ones that are known, viruses that are known to cause cancer. And 10% of cancers, about 10% of cancers are known to be virally induced, meaning that they were actually brought on because of a viral infection. So, question for you. All of the following can be used to culture animal viruses except, so is it red, laboratory animals, yellow, cell culture, green, embryonated eggs, or blue, glucose broth? So go ahead, think it over, push pause, and when you're ready, go ahead and turn it back on. So if you said glucose broth, you are correct. Again, remember, viruses are obligate intracellular parasites. They have to live within a cell. So you can't just grow viruses in a nutrient broth or a glucose broth because without cells, they're not going to be able to replicate. Remember when we did our plaque assay that in order to replicate or to be able to amplify the virus, we had to also give the virus particles E. coli, which are their host cell. So the ones that we can use to culture animal viruses, laboratory animals, cell culture, and embryonated eggs. All three of those allow the virus to be replicated within living cells. And so we're going to look at viral multiplication of bacteriophage. So bacteriophage are viruses or phage that infect bacteria. And one of the things that you're going to notice is that the multiplication of these bacteriophage is very similar to animal viruses. They have many of the same steps. However, there are some distinct differences as well. In a phage, we have our protein capsid head, and within that is the nucleic information, in this case the DNA. It has the sheath and it has the tail fibers. And so remember that in the animal cell multiplication, the capsid actually enters into the cell and then undergoes uncoating. In phage, though, the capsid never enters the cell. That's because the phage lands on the E. coli and it injects its DNA into the host cell directly, meaning that the capsid never enters the cytoplasm. And so in this case, uncoating is not necessary because capsid does not enter the cell. Additionally, the way that the virus is released is different for phage versus animal cells, in that for phage, release only occurs by lysis only. Phage do not do budding, and that's for two reasons. One is if you think about bacteria, bacteria have a cell wall, whereas we don't. So to do budding and to do exocytosis, remember that in budding, the membrane starts to bleb out and the virus takes the host cell membrane with it. If you have a rigid cell wall, it's much more difficult to do that. Additionally, notice that for the phage, that they don't have an envelope structure. And because of the lack of envelope structure, that tells you that they don't do budding, because the envelope comes from the cell membrane of the host. So just like animal viruses, bacteriophage must be grown in living cells. And the living cells in this case is going to be bacteria. And so you guys saw this in lab when you did your plaque assay. And what we did was we took our E. coli and we mixed it with the virus particles, and then we poured plates. And remember that anywhere that the plate was cloudy is a lawn of bacteria, meaning lots and lots and lots of bacteria. And viruses are indicated by these plaques. And these plaques are these clearings. And why this agar is going to be clear versus cloudy is that wherever you have these plaques, that was one virus that infected one E. coli. That virus turned the host cell into a virus-producing factory, and then the E. coli lysed. Virus went on and infected adjacent cells. Those lysed, infected neighboring cells, and so these clearings are basically places where the E. coli have lysed open. And we call these are referred to as plaques. Each plaque corresponds to a single virus particle in the original solution. And these can be expressed as the plaque forming units, meaning the number of plaques on the plate, which can be used to determine the titer of that particular phage. And again, that's because each clearing is the result of one virus infecting one E. coli, lysing it, and keeping the process going. So, if we look at phage multiplication, so we have attachment, and in attachment, this is where remember that the phage is a complex virus. Here's its capsid head. Here are the tail fibers. Tail fibers will land and attach at the E. coli or the bacterial cell wall. And so this is when attachment happens. The tail fibers attach to the host cell. Then we have penetration. And during penetration, remember that I talked about that one of the enzymes that some viruses have is something called lysozyme. And lysozyme is an enzyme that's used to degrade cell walls. And so in this case, the phage produces the lysozyme, and it allows the virus to be able to break down the cell wall. And that's important because once it's able to penetrate the cell wall, this sheath contracts, and that then leads to biosynthesis, meaning that now the DNA is injected into the cell. And now during biosynthesis, this is again similar to our synthesis. So we have our production of phage DNA and proteins. So it's going to replicate all of its parts. Then we have maturation. Maturation is the same as assembly. This is where we get assembly of the phage particles into virions, which remember are the complete infectious virus. So we have to put all the parts together. So notice that during maturation, it's going to start to assemble the virus particles. And then lastly, we have release. And again, lysozyme is produced, and that's used to break the cell wall and release the virus particles out of the cell so that now they can go on and infect another bacteria and keep the cycle going. Notice that during this step, there is no uncoating step. Notice there are 5 steps versus 6 for the animal cell multiplication. And that is because, again, the capsid never enters into the host cell. And that's in contrast with an animal cell viruses. So, if we're looking at phage, adsorption and penetration. What's going to happen is that the phage are going to use these fibers at the end of the tail, fibers, to attach to cell wall. Then the phage produce lysozyme to degrade the cell wall, to degrade bacterial cell wall. And then as it breaks down a portion of the cell wall, this tail fiber or this tail sheath contracts, and when it contracts, this tail core is driven down into the cell wall. When the tip of the core reaches the plasma membrane, the phage DNA is going to pass from the head, the capsid head, to the tail through the plasma membrane, and it enters into the bacterial cell. And so this is going to be adsorption and penetration. It's going to attach using these tail fibers, and then it's going to penetrate by using lysozyme, breaking down the cell wall. The tail sheath core then is injected into the cell, and now the viral nucleic acid enters the bacterial cell. And so what you're looking at here, we have an electron micrograph, and you can see these phage attached to the outside of E. coli, and you can see that they're at various stages in their multiplication, meaning that in this case they've injected their DNA, and they're going to use the virus, or they're going to use the bacteria as a phage producing factory. And so again, release of the phage happens through lysis. So occurs by lysis only. And again, they can't do budding. And so what's going to happen is that once enough virus particles accumulate in the bacteria, the bacterial cell is going to rupture, and it's going to release all those virus particles into the surrounding area, where they can then infect an adjacent bacterial cell and keep the process going. And again, we can detect this by using a plaque assay, looking for regions where lysis has occurred. So for bacteriophage, a particular type of phage called a lambdaphage can do something called the lysogenic cycle. And what that means is that lysogeny is where the phage remains latent. It's not actively producing more phage particles. It's almost, think of like when we talked about animal viruses. Remember, we talked about latency, where they can hide out and they don't make new particles. In phage, we call that the lysogenic cycle. And so what happens in the lysogenic cycle is that the phage DNA actually incorporates into the host cell DNA. And this inserted phage DNA is then known as prophage. When the host cell replicates its own chromosome, it also replicates the prophage DNA. And this then results in something called phage conversion. The host cell exhibits new properties. So if we look at the lysogenic cycle has where, again, we talked about that the phage DNA, when the phage injects it, can circularize, and at this point the cell can either go through the lytic cycle or the lysogenic cycle. And if it's in the lysogenic cycle, the phage DNA will recombine with the bacterial DNA, and that will then be the prophage. And so again, anytime that this cell replicates, in addition to replicating its own DNA, it will also replicate the prophage DNA as well. And so this lysogenic bacterium reproduces normally, and occasionally the prophage may excise or be cut out from the bacterial chromosome by another recombination event, and that then frees up that phage DNA, and now that cell could go back into the lytic cycle where it's actively making new phage particles. And these types of phage are also referred to as temperate phages. And that is that they don't necessarily cause the death of the host cell. They might just be waiting out until conditions are favorable again. So bacteria may acquire the ability to produce toxins when they're infected with lysogenic phage. And so if you remember back to when we talked about horizontal gene transfer, that one of the ways that DNA sequences can be transferred from one organism to another is through, again, transduction. And in transduction, cells use a phage or a virus that infects bacteria, and that phage sometimes can pick up some of the DNA from a particular bacterium and then carry that DNA to a new type of bacteria, and that DNA can be incorporated then into the recipient's DNA. And so an example of that is E. coli O157H7. Remember that E. coli is normally found in your gut, no problem. However, if you happen to get an infection with E. coli O157H7, that happened because of lysogeny, meaning that in this case, E. coli picked up the ability to produce the Shiga toxin. It got this gene from Shigella, and when it has that lysogenic phage in it, it allows it to produce that Shiga toxin, and that's what leads to that very severe diarrhea, etc. For Streptococcus pyogenes, it can produce an erythrogenic toxic. And if it produces this toxin, that can lead to scarlet fever or toxic shock syndrome. Corynebacterium diphtheria can cause or produce diphtheria toxin. Clostridium botulinum can cause botulinum toxin. Staph aureus can cause toxic shock syndrome. And it causes these diseases, or causes the bacteria to produce these toxins when they are infected with the lysogenic phage. And so if we compare phage and animal viruses, so looking here, so here we have bacteriophage and animal viruses. They both have attachment and entry, but again, the big difference being that uncoating does not happen in the phage. So let's talk about the way that these things occur. So in a phage, again, attachment happens when the tail fibers attach to the cell wall of the bacteria. Animal virus, the attachment sites are on the plasma membrane through receptors, and this can recognize specific proteins or glycoproteins. In terms of entry, in phage, the viral DNA is injected into the host cell, whereas for animal viruses, entry or penetration occurs by either endocytosis or through fusion of the virus with the cell. Uncoating is not necessary in phage because the capsid never enters. However, animal viruses, the capsid goes in, and so they have to have the protein capsids removed to expose the nucleic acids. We have biosynthesis, and in biosynthesis, the cell is going to replicate nucleic acids. It's going to make more parts for the phage. It's going to make the protein capsid, etc. In bacteriophage, this happens in the cytoplasm. In animal viruses, if it's a DNA virus, this will happen in the nucleus, or if it's an RNA virus, that'll happen in the cytoplasm. Excuse me. We could have a chronic infection. And a chronic infection in using phage is called lysogeny, again, meaning that they're in that lysogenic cycle. They're not actively making new phage particles. In animal cells, that is referred to as latency. And so during latency, again, the virus is inactive. It's dormant for a period of time, but it might reactivate, and this then could lead to new infections. Chronic infections could also be persistent, meaning that the virus particles are produced in a very slow manner, so that would be slow viral infections. This also could lead to cancer, meaning that the host cells may change in such a way that the host cells then have the ability to replicate out of control. In terms of release, again, this is one of the difference. In phage, the way that the virus is released is by lysis, whereas for animal viruses, there's two ways through which they can leave. If we're talking about an enveloped virus or a virus that has a membrane on the outside, the way that it's released is through budding. But if we're talking about a non-enveloped virus, then it's going to use lysis, or rupture of the host cell, in order to release the virus particles. And so that's the difference, is that animal viruses can use budding, phage cannot. And so now we're going to look at prions. Prions are proteinaceous infectious particles, meaning that in the case of prions, prions are not actually like a virus. They are simply misfolded proteins. They have no nucleic acids. They are just these proteins that would normally be found, but they are misfolded in such a way that they cause disease. And remember that for prions, that they're extremely resistant to usual sterilization techniques. Remember that these are the most resistant to antimicrobial chemicals or to other methods for sterilization. And again, that's because we're not targeting something living. These are simply misfolded proteins. And so these prions can cause transmissible spongiform encephalopathies, which are these basically fatal neurodegenerative diseases. And these types of infections are common in animals. It can cause something called scrapie in sheep and goats. It causes bovine spongiform encephalopathy, BSE, or mad cow disease, right? You might remember back a while ago, there was an outbreak of mad cow disease from Jack in the Box, and it came from eating meat that was not cooked enough and transmitted these prions from the cows to people, and this was a very serious disease. It can cause something called wasting disease. In humans, it could cause this Creutzfeldt-Jakob syndrome, which is actually thought to be there's thought to be a genetic component to this one, meaning that it's thought to be able to be passed down from person to person. And so the way that mad cow disease arose was from feeding the scrapie infected sheep meat to the cattle, and then the new bovine variant was transmitted to humans who ate the undercooked beef from the infected cattle. And so this can these can be transmitted um, through various mechanisms. So there are other non-cellular infectious agents, what are called satellite viruses, and these viruses are dependent on other viruses for replication, meaning that they can't infect a host cell by themselves. There has to be an additional virus in that host cell with them in order to replicate. An example of this would be adeno-associated virus. These can only replicate in cells that are infected with the adenovirus. And again, adenovirus causes respiratory infections. There's a satellite virus called a delta agent, and this is where naked strands of RNA are expressed only in the presence of the hepatitis B virus. So only if hepatitis B virus is present will this occur. There are also what are called virioids. These are short pieces of RNA. They're no protein coat. These have only been identified in plants so far. And so this concludes our virus lecture. So the last question is, viruses that infect bacteria are called red bacteriophage, yellow prions, green virioids, blue antisense viruses. So pause when you're ready, turn it back on to hear the answer. So if you said bacteriophage, that is correct. Those are the viruses that infect bacteria. It's not prions. Prions are infectious proteins. It's not virioids, which are naked RNA sequences that don't have any protein components. And it's not antisense viruses.